To start, do you want to just introduce yourself?
Sure. So my name is Shih, and I'm an assistant professor here at Concordia in the Department of Electrical and Computer Engineering. I'm also cross-appointed with Department of Biology. My degrees are in electrical engineering, chemist. I have a master's in chemistry, and also a PhD in biomedical engineering. So I would say I have a very sort of interdisciplinary sort of background. Yeah.
Okay, hi, my name's Dr. Christopher Brett. I'm an associate professor of biology, and my lab studies basic cell biology, fundamentals of cell biology, and we're starting to get into synthetic biology too.
Would you describe yourself as a synthetic biologist?
Not in the fullest sense. I use synthetic biology as a tool in my research. Mm-hmm. So one of the things that we need to do is, say, modify genomes, and we would use synthetic tools to do that.
Would you consider yourself a synthetic biologist as well? Is that a term?
Yeah, I would say so. Yes. It's such a broad term. It's anyone that sort of engineers an organism now is sort of considered like a synthetic biologist. So, if you play with cells, you play with proteins. I think everyone can sort of say that they're sort of a synthetic biologist. But for me, like when I consider a synthetic biologist is someone that definitely engineers, some sort of organism and at the same time produces something valuable. I think if they get to sort of that stage, I always consider them as, they're the true sort of synthetic biologist. Yeah.
I think that's a fair way of describing it. It's sort of it's a very general term, and it's kind of a very interdisciplinary field. There's a lot of diversity in the kinds of topics of research, but it's all coming together. Biologists, chemists, engineers, to engineer biological systems and to try to use these synthetic biology approaches to do useful things.
How do we describe synthetic biology? So it's funny that you say that, because I would say that I have probably been incorporating synthetic biology in my research for my entire research career without realizing I was doing synthetic biology. So it's really interesting. It's an interesting question. I think I see it as taking information or material that may exist in one type of system or organism and then swapping it maybe into a different system or organism, or modifying an existing material within a system, creating new material within a system, and then using that in a way to understand fundamental functions. So understanding how that cell or system or organism actually works.
I think even synthetic biologists don't really know what the definition is. It's kind of a carryover of bioengineering, which has been around for longer. I think everybody will tell you that. But it's meant to be a combination of engineering and studying or manipulating, finding applications by applying engineering to nature or biology specifically.
The synthetic aspect of this work is that you can now engineer not just one gene, but entire systems of more than hundreds of genes, or tens and hundreds of genes in a single cell.
Basically, there's kind of limitless applications. It depends on basically our creativity at this point. Scientists have spent years creating tools to do it, and now they're kind of finishing what are called proof of concept experiments, basically figuring out, okay, can we get this to work? And now they're applying it to all sorts of weird stuff, like a good example is, Beyond Meats, making synthetic meats, things like that. So they're worried about, say, food security. I know there's companies in Boston making using synthetic biology, making fish substitutes now instead.
Yeah. Actually, so when I did my postdoc, that was really when I first got exposed to synthetic biology. And the first one that always comes to my mind is biofuels. That's the one that I work with a lot. And so the idea is that, having something more green, something better for the environment. So the idea is that you would engineer a cell or some sort of pathway inside a bacterial cell or a yeast cell such that they can produce any sort of these green or advanced biofuels. The idea is that they would have the same chemical properties as you would see at a Petro-Canada or Ultramar, and now just cheaper and better for the environment. Unfortunately, that's still very hard to do, but there's been so much progress, and so there's nothing yet in the market. But some of the, I guess, real life, market examples, one that always comes to my mind as well is drugs.
I'm David Kwan. I'm an assistant professor in the Department of Biology, and I'm working in the Center for Applied Synthetic Biology. So I guess I could give you an example where we're talking about engineering microorganisms to produce medicines. We use a lot of medicines, actually. A lot of the medicines that we use do come from microorganisms, from bacteria. It's particularly true for antibiotics, for example, but it's also true for some anti-cancer drugs. And sometimes we use these as they come from the microorganisms, or sometimes they need to be chemically modified. And they're sort of conventional medicinal chemical approaches that you take these molecules and you do some chemistry, and then you get the drug that you're using at the end. So there are some examples of anti-cancer drugs that normally the molecule that you get from the microorganism, it's an anti-cancer agent, but it has some side effects, like it after prolonged use causes kind of heart conditions, that sort of thing. What we're interested in is, can we engineer the enzymes, or can we engineer the molecular machinery within these microorganisms to do that chemistry for us, do that on their own. We have kind of broad interests in my research group, but I guess an overarching theme is that we are interested in studying the function and engineering of enzymes and enzyme pathways. Well, I could say it another way. We're focused on how enzymes and enzyme pathways operate as the molecular machinery inside cells, and we're trying to, in some cases, engineer that. We're trying to tinker with that machinery.
So my name's Alisa Piekny. I'm an associate professor in Department of Biology, and I'm also Tier 2 Concordia University Research Chair in Cancer Cell Biology.
So your research, if I understand correctly, and I may not, focuses on cell division and shape changes to sort of discover the process especially unique to cancer cells. Is that a fair?
Yeah, so I would say, just to modify the question a little bit, it's that we're studying those things so that we can perhaps develop tools such that could be used as therapies, or applications that could be used in a therapeutic way to help, say, treat cancers. And so cancers, yes, two of the, we call it hallmarks, are uncontrolled proliferation and ability of cells to migrate from their primary location to secondary locations. So we're trying to understand the fundamentals, again, the fundamental processes underlying those things. And how they go awry in cancer cells. So then once we understand that, now we have something to target more specifically. And so part of my program has actually gone there in terms of the application side by partnering with medicinal chemists, and now we're designing compounds to specifically target components that are involved in helping to regulate cell division, for example. And so we can do this in a very targeted way. And by understanding the biology and marrying it with the medicinal chemistry, we can come up with drugs that are very highly targeted and hopefully will be more successful in the clinic, but let's see. In terms of the tools in general that we use in our research, we do things like genome editing, right? So we would use CRISPR, for example, to introduce a fluorescent probe into the genome so that we could visualize where something is locating within a cell. We can use it to generate mutations in specific locations so that we can understand, again, how that particular component works, and in a very specific way. We would use microscopy, and we use quite a bit. That's my primary expertise, is more on the microscopy side, but we would use things like confocal imaging, turf microscopy. These are all fancy terms that nobody would know, but what it means is that we can achieve high spatial resolution within a very, very small space. like a cell. So we can see very precisely where things are going, and more importantly, when they're not where they're supposed to be, right? So other tools that we might use are more biochemical techniques and in vitro, where we can look at how things interact together. And that's how we design the mutations that we make in the cell. So we look in the test tube and we see how things are working or not working properly. That guides us to where we should be mutating within the cell so that we can perturb those things more specifically. So yeah, that's it in a nutshell, I guess.
Awesome and complete answer. And all of that happens here in this lab?
Yes, so I mean, here is where we do biochemical work, we do molecular work, but of course we need to do a lot of work in cell culture space, so that we have upstairs, we have a big room that's dedicated for cell culture use, and this needs to be a level 2. It's a protected space because we need to take precautions, of course, when working with these cells. These are human cells. We also have microscopy rooms, so these are dedicated rooms where we have the microscopes, again, they're quite sophisticated, and they require dark to be used properly. They require low vibration, so we have to have dedicated spaces for those too. Yeah, so, and then we have the foundry, of course, which you should know very well by now. And so this has become a very important tool for us as well. And so when we want to screen, for example, when we edit cells, and we want to screen large populations of those cells, or when we treat them with cancer drugs, we need to screen large populations. Yeah, we need to use the foundry for that work.
And that's a space pretty unique to Concordia.
That's very unique to Concordia, yes. And so the ability to do something like that combined with the other work, which I would say is maybe more traditional for many universities, I think that's what makes us stand out.
In medicines, it's kind of the classic has been reprogramming people's immune cells to attack cancer. That's one of the first kind of synthetic biology technologies or applications that have been applied to medicine.
So the first ever sort of synthetic biology example that gone from sort of the research lab to the actual industry and people can actually use it is essentially this drug called artemisinin, which is an anti-malarial drug. Before, the way that people have been using this drug, the dose per, I think it's like $100 per dose or even higher than that. But some synthetic biologists, they were really smart with that, and they were able to essentially replicate the pathway implants and then place that pathway inside a yeast cell, and then essentially be able to produce artemisinin through yeast cells.
And so the idea is we can reprogram bacteria or yeast or pretty much any organism, but it's mostly bacteria and yeast, and make really rare or hard to synthesize chemicals, bring down the cost of manufacturing. And so those are some of the applications people are using now, biofuels, things like that, reprogramming, say, plants, or even, blue-green algae, and then so they kind of power the synthetic, whatever process within them that has been built, using light energy, for example. There's all this weird stuff in terms of reprogramming cells to respond to electricity, too. So to basically create these machine biology interfaces, things like that. Tissue development in vitro, like making fake skin, right, for burn victims, things like that. Eventually grow out of people's stem cells whole organs in vitro, so you could have an organ transplant with your own heart that was, replacement heart that's grown outside of your body and put back into you, are some of the real goals that countries are trying to accomplish, including our own. So, or things like the universal stem cell. Could we make a stem cell that we can make all these things in and then put them into any patient, any person if they need some sort of therapy that requires, they're called cell therapies or gene therapies. So that type of stuff.
We're ready.
Hi, my name is Aashiq Kachroo. I'm a professor at Department of Biology, Center for Applied Synthetic Biology at Concordia University, Montreal. I'm a recent recruit. I joined in 2017. My research at Concordia University focuses on genes that we share with other organisms, including humans. And we ask a very simple question of whether these shared genes, especially from humans, can swap or be replaced in simple organisms like budding yeast, You also know this yeast as a baker's yeast, which we use in our bread.
How do you relate to the word synthetic?
Yes, it's an excellent question. So the way I relate to synthetic biology, the current projects that we have in the lab, is the following. What we're asking is, we basically tested the genes one gene at a time. We said, okay, we'll not delete one of these genes, but take the shared gene from humans and test whether it functions. And we discovered that this swap or these replaceabilities were driven by a module or a network of genes, not just one gene at a time. And thus realized that it's possible not just to swap one gene, we can in principle swap the entire processes inside the cell. And so the synthetic aspect of the work is the following. Can we now take the entire system from the yeast cell and make it human altogether?
And this is where things get a little crazy. We're taking human genes and we're expressing them in yeast cells, because all cells, including yeast, make these exosomes for communication. Manufacturing the DNA is the toughest part, I guess, right? And then after that, you just treat the yeast with it. They can't wait to take up the DNA. It's part of their way they share genetic information. And then through what's called homologous recombination, they just take the foreign DNA, you pour it on top of them, and we'll integrate it into their genome.
And so the reason why we are doing this is the following. Why do we swap genes? What is the point of this? We really ask very fundamental questions this way. Can genes function the same way or not? That's a really critical question. Number two, these yeast cells, which now have human genes in them, acts as test tube for studying that human gene, particularly the gene that you swapped. You can mutate that gene. You can create mutants like disease variants in that gene at a scale that you can do in a simplified cell, like thousands of them at a time. And so that directly relates to what that mutation does to the human gene. It might cause disease, and we'll know why it causes disease because we studied in a simplified cell.
If you can imagine a patient that has a mutation in a very specific gene that's causing that particular disease, imagine if we could go in and edit that, right? We could actually cure somebody of a disease rather than treating them just for the symptom of that disease. Another way that I see the future of synthetic biology heading is, say you need to have a liver transplant. Well, right now that's very challenging to do. You need to match donors really well. Well, imagine if we could grow a liver in a lab that's pre-matched, right? So now you're not waiting for a patient to match and then to get that, to do that particular transplant. So I could see it being transformative in health.
Looking at genetically gene editing microorganisms, this can be useful for making sort of microbial cell factories, and there's a couple projects here at the center that I'm involved in, along with some collaborators. So my lab in particular is interested in kind of manipulating and engineering the molecular machinery within cells, proteins inside cells. But part of the larger picture is to engineer these cells. A lot of the times it's yeast to produce useful things, and some of these things could be medicines. You introduce the genes into these yeast, and then they'll produce these molecules that we use as medicines.
You're right. Many people may not know why we are studying budding yeast in the laboratory. Budding yeast has been as a model for fundamental science for like decades now. The reason is because it's a very simple organism to work with. We know a lot about this organism genetically and phenotypically. We know how to grow it. We know how to manipulate it easily. So the tremendous model organism. We have lots of resources for this to work with this organism. And more importantly, we share genes, especially with humans, and significant number of them. Like if you look at the number of genes that human genes share genes in yeast, we are looking at around 4,000 human genes, all important for critical functions in the cell. So clearly an amazing model because of its similarity with humans. So many biologists know this for a while now, that we have shared genes, especially between humans and yeast. So we asked a very extreme question in terms of you completely remove that gene from this baker's yeast, and then swap it with the human version of that gene and ask whether the human version will cover for the function of the deletion of that yeast gene. And so we did this in a large scale and found many of them. Almost half of the tested ones were able to do that function, even if they are a human gene. So clearly they are not only similar, they also do exact same function.
So I came up with an idea a while ago that seems to be liked by quite a few, and there's a few companies that have started doing this, but it's basically the ways we're all made up of cells. All living organisms are. That's the smallest unit of life. And so bacteria are single cells, yeast are single cells, and humans are made of trillions of cells, like a big cell community. And the way they communicate with each other is through something called one-ways they communicate. And I think everybody knows that's one-ways, hormones, for example, things like that. But they send little packages to each other. They're called extracellular vesicles or exosomes. And inside of those packages, they have things like DNA, well, mostly RNA, proteins that they can share between the cells. And it's used as a major form of communication between cells. And this has only become realized recently. And so people are now starting to use that for, say, diagnostics. Let's say if you have a certain type of cancer, those cancer cells will communicate using these packages, these exosomes. And they actually release them into your blood. They get caught in your urine, okay? So they're also in your saliva for cells to communicate, different cells to communicate. And so they're used, for example, as diagnostics. So a cancer cell will release an exosome with a certain signature. And if we can detect that, we can use it to say, oh, this person might have this type of cancer. But we want to reprogram them, basically, so that we can use them to fool cells. We can hijack this system and come up with ways that cells would normally communicate. We can hijack that and maybe make them have alternative decisions. For example, cells communicate when they're growing into a tissue. Like as you're developing, as the heart develops, all the cells have to coordinate and they send exosomes to each other. Okay? And by doing that, that allows them to form a heart. So if we want to realize these goals like making someone's heart in a petri dish, right, for a transplant or things like that, we need to come up with a way of synthetically making these exosomes that could, say, trigger that or help that. There's a lot of other things you can do too in terms of this would be called something like a vector for gene delivery. So because we can basically put inside of these packages whatever we want. And what's another cool thing about them too is once a cell releases an exosome, it targets only another type of cell, right? So for example, a cell in your brain can target another cell in the brain very specifically, or it'll just target, let's say, a liver cell or something like that. And one of the big problems confronted with, say, the pharmaceuticals industry and drug making is we've had to shelf a lot of drugs because they do not target the right tissues properly. So they affect all the tissues in your body, and that's what leads to side effects. Some of them are so extreme that the cost, the deficits, the side effects, if they're too bad and they overwhelm the person, it overrides, right, the therapy itself. So if they're too toxic, for example, those things are shelved. And there's been a lot of drugs in development over the last few decades where they can't just target the drugs. So the idea is maybe using something like exosomes, you could put the drug inside of them. You can have the targeting signature on them to say only go to the liver, only go to the brain, only go to places like that. By coating them in this package too, right, your body assumes it's part of you and it's not foreign. So it doesn't try to destroy it. So it's a way of kind of protecting whatever you put inside.
So these are beneficial tools that we've been using for many, many years. We may have been doing it in a different way where we've been doing a more classic genetics approach, but in that approach, we're modifying genomes. Mm-hmm. So when we use CRISPR, this fancy new genome editing tool, well, it's also editing genomes, just like the way we were doing genetics before. It's just this way it's a little more targeted. So we can specifically mutate the DNA at a desired site rather than hoping to get it at the site that we're looking for.
Not a new thing, but what we did was we did it at a systems level. We just didn't do it one gene. We took an entire set of genes and asked what happens if you look at hundreds of the genes, and we might come up with the properties of these genes which are critical for them to be, swappable in a different species. So we did end up, that was the rationale for doing this at a systems level. So the rationale for systems biology, systems and synthetic biology now in my lab is that can we now replace systems instead of single genes? And this involves genome editing, genome manipulation at a very large scale, and simultaneously asking questions of how, can we now can these systems as a whole function in the context of this simple organism?
I mean, there is this weird stigma attached to it, or any sort of biological engineering, like GMOs, genetically modified foods and things like that. But the reality is, you know, most of the products that we do use are derived from either some sort of synthetic process, chemical synthetic process, like drugs, or they're derived from, you know, some sort of genetically modified animal. It's just kind of taking it to the next level to build kind of new things that would be useful for society.
Destigmatize synthetic biology. I think that a lot of the problem is that there's a lot of disinformation or misinformation, a lot of misunderstanding of synthetic biology, and there's sort of a saying that a lie makes it half the way around the world before the truth has a chance to get its shoes on, right? So there's the challenge to kind of counteract, you know, the misinformation that's out there.
I mean, even just talking to my mom, I think, you know, when she sees the news, you know, like she says like, oh yeah, you know what? I hear China, like they can use something like this CRISPR protein, and then they can make like different sex babies and babies with a preferred sex. I mean, when they hear that, then they say, oh, you know, when you genetically modify something, that's a bad thing. So I think the main thing is that you have to educate. I mean, education is just utmost important. So when I hear that from my mom, I'm like, yeah, I mean, I think it's our duty and any sort of people that are working in this field to essentially create, I don't know, conferences or workshops that are essentially open to the public, and then such that we can essentially educate them.
I think what maybe scares people is the power of this method, right? Being able to genetically manipulate all organisms for a certain purpose. You know, it could, in general, I mean, it could be used in the right ways and used in the wrong ways too. I think most people who study synthetic biology are trying to, you know, figure out a problem or come up with an application that's useful for society.
Maybe the best way to go about it is to emphasize that it's an important thing to get right. So I can sort of give you an example of something that's been in the news recently, in the last couple weeks or so. There's a story of golden rice. Across Asia, there's this problem with vitamin deficiency, and there are hundreds of deaths per day as the result of vitamin deficiency in poor communities. Lots of children are suffering. And recently, this technology of golden rice, editing the genes of rice crops to introduce vitamin precursors for them, that has now the potential to save hundreds of thousands of lives per year. And this is an example of synthetic biology. But the incredible thing is that we've had this technology since 1990s, right? We haven't been taking advantage of this because of this stigma, because of this kind of disinformation and this kind of this irrational fear of genetically modified organisms. And you know, you think about the millions of lives that could have been saved over this time from this technology and technology like it, right?
So when we have these sort of conferences, we tell them like, this is not what we want. We want to use synthetic biology to essentially create new drugs, drugs that can help the poor that can't afford it. We want to essentially engineer food that for places that can't agriculture their own food or don't have the animals to produce the meats. And so these are the things that we want to push for and educate them that there's really essentially no health defects, or there's essentially all benefits.
And this is where the public, I think, needs to understand that we realize that there's still a responsibility with how the research is being developed. And so there's policies in place to help ensure that we are responsible and how we're moving forward with this research, right? So that it's done in a very ethical way. So I would say that that should alleviate stress for the public to know that we are being socially responsible.
So I think, it's always about just standing up for what you believe in and then educating them the right way, because maybe they just don't know. So now I would say my mom can eat that, I guess, strawberry that's been genetically modified without any worry. So yeah.
There's a potential for real benefit and real progress with this kind of technology.
I mean, my research is obviously in the health sector, so I'm really using these tools to understand fundamental processes, say underlying how a cell divides, and then by understanding that information, we can generate drugs that target that process and that can be used to treat cancer, right? So I think in terms of the benefit of synthetic approaches, I see it from my eyes. I can understand the health benefit the best. I think if you, again, let's go back to the genome editing example.
When we deal with multiple number of genes and the data that we get, it's really hard to do it by conventional means, which is by simple looking at a gene and physically looking at the computer sequence of that protein. And it's very hard to do it with hundreds and thousands of genes. Therefore, we really rely on computational methods that can predict and do this through computers, the predictions of what we are trying to observe in our results. For example, one of the first studies that I did, we looked at over 500 genes that we can swap from humans in yeast, and then we verified, quantitatively tested the features of these genes, 104 different quantitative features. It is not possible, by conventional means. So we use computational pipelines to understand what each feature tells us about that human or the yeast gene that we swapped. And now the scale is going in higher. We now have 700 plus genes. And now we're trying to do this in a different yeast, which again involves hundreds and thousands of genes. So without using computational pipelines, it's just impossible to come up, with the results, the analysis of these results without doing computational pipelines.
Maybe I'll give you some background. Like Microfluidics is essentially integrating essentially this lab that you see here onto sort of this credit card device. So essentially it fits right in the palm of your hand. And if you think about, I know when I was in high school, I always did these experiments where, you had all these beakers, right? And then you try to essentially, mix A with B or mix C with A, things like that. So essentially what we want to do is sort of remove that sort of manual intervention that, you have to sit there and watch it and then do all the manual pipetting steps that are involved. Essentially what we want to do is integrate all that in sort of an automated fashion right onto this credit card size device. And the whole point, the main, I guess, sort of advantages with microfluidics is now we can automate. So we have this sort of box here, which essentially is connected to the device, which will essentially automate all the sort of fluid handling steps that are necessary in any sort of chemistry or biology experiment. Now, since we're taking the lab scale, reducing it down to, the palm scale, now we're looking at volumes that are 1,000 times less. So not only do we reduce volumes, we also reduce the costs, significantly. And just miniaturization just brings so many benefits in terms of multiplexing. And so what that means is that we can essentially perform multiple experiments, hundreds of experiments at sort of one time in parallel. So here I show you just a device where you can probably do maybe not 100, but you can do a good number like 16. And I know when I tell my students that, hey, why don't you guys do 100 experiments today? I mean, if they didn't have microfluidics, I think they would sort of freak out. They'll be like, I'm going to be here all day, all night. So having that sort of miniaturization, having the possibility of multiplexing just really helps. And one of the projects that we're really sort of excited about in terms of using microfluidics is the idea of cellular therapies. And the reason why we think microfluidics can really help in this sort of area of synthetic biology and essentially this area of cellular therapies is that now we can place essentially the cells that we want to engineer all onto this platform. And the beautiful thing is that because we can miniaturize, we're now able to handle some of these sensitive cells or precious cells that we're not usually able to handle with standard tools. And so now we're just, the handling that we have, we're just inducing less stressful forces, I guess, onto these cells. So now we see just a better or more or different effects on these cells. And because of that, we can then re-engineer these different cells and then be able to essentially use synthetic biology and automation to produce cells at a much quicker rate or engineer cells at a much quicker rate. So I think, I guess if we had to sort of sum it up in sort of a nutshell, I think the ultimate experiment for us would be to essentially place, this system here, it essentially contains, a computer, this device holder, and this automation setup right here. And the idea is that we can place this in a doctor's office, right? A clinical sort of research lab, even just my collaborator beside me. And the idea is that, we would take a patient's blood and then be able to put it on to our device. And then they would re-engineer the cells that would, kill off cancer cells or make them more precise, that would attack exactly those cancer cells. And then after maybe a couple days, we would then take it back, now we've engineered them, and then place it back into the patients. Exactly. And then essentially, we just want to democratize essentially cellular therapies. I mean, now this would be available in all the offices, and maybe we can reduce waiting times. That'll be the best thing. I wait so much time at the doctor's office sometimes. So yeah, this would give opportunities for not just one hospital, but maybe multiple hospitals, or just the clinical office down the street to be able to do these sort of tests, and then to be able to give patients what they deserve, especially these cancer patients that don't have possibilities of chemotherapy or other types of therapies. So this is one project that we're really excited about, and I think really microfluidics can really push through that no other platform can. So, yeah.
But so we can make thousands of these. We'll make thousands of strains, making thousands of different exosomes, and we can kind of, when you have this opportunity to try so many, right? We need things like engineers to help us decide, okay, what's the best approach for this? Or we have to use, say, we have to mine the literature with, not AI, but at least some computational methods to figure out, okay, what could be the players? What are the human genes that are important for these different effects? So that's how we integrate, right? The computational aspect, the engineering aspect.
Yes, absolutely. We have collaborations with people who have computational tools, who have developed computational tools for years. Without those, it just is not possible for us to, come up with the analysis and find interesting features of the data without using those tools. It's just not possible. So we need collaborations with experts in the field of computational bioinformatics specifically.
I would say the hardware side of things, in terms of the automation system, I would say we can. So we already have some of these systems in some of our collaborators. So I have a collaborator at UDM, University of Montreal, and they already use some of these systems already. Now, if we were to put it into a doctor's office, for sure there's still a lot of work in terms of the biology and in terms of, do those cells actually do what we engineered them to do. So there's definitely a lot more testing. But if I had to give a number, I would say definitely within the 5 to 10 year mark. we can definitely have something like this. I mean, there's already, in this area of cellular therapies, there's already one type of engineered cell, or these specialized, what they call T cells, specialized T cells in sort of clinical trials. The only thing that's just only one type, and it's really difficult to make many different types. So I think once we have this, then it will just expedite the process so much more. So yeah, I would say 5 to 10 year mark. I'm sure someone that hears me saying that, they're like, no way. But for us, I really do think 5 to 10 year mark is very possible. So synthetic biology is just growing so fast, and so we're learning so much. And again, I think, luckily I'm not doing the work, because if I was doing the work, I probably won't be 5 to 10 years. I mean, luckily it's my students. They're really fast with, so I believe, yeah, 5 to 10 years for sure.
I don't think we're that far. I mean, you know, this idea of genome editing in precise ways, or that, and understanding, I guess, how networks are controlling some of these pathways that would mediate things like, say, tissue rejection. I think combining those tools, right, is something that we never had the ability to do before. And so now I think we're at a place where that doesn't have to be that far in the future. But again, as soon as we talk about biology, it's very complex.
Right.
And so I can't predict how far in the future that would be, because for everything that we understand, there's about 1,000 things we don't understand. And so we could still be far away for reasons that we really don't know yet. But I would like to say that we're closer than we've ever been.